Eukaryotic transcription. The process in eukaryotic cells is a bit more complex. The process still consists of the same 3 steps, but each of them has complexities that makes transcription in eukaryotes a more difficult process. The first difference is that eukaryotes have more than one RNA polymerase. Each of the RNA polymerases in a eukaryotic cell is responsible for transcribing different subsets of genes. The polymerase that we'll focus on in this class is RNA polymerase II, and that's responsible for transcribing all mRNAs as well as all microRNAs and some heteronuclear RNAs as well. This image very clearly shows that the process of transcription initiation is much more complex in eukaryotic cells than it was in prokaryotic cells. There are, however, some similarities. For example, transcription will be initiated at a promoter site, and that promoter site will be upstream of a gene immediately adjacent to it. In addition, that promoter site will contain a TA-rich sequence that we'll also call the TATA box. However, in a eukaryotic cell, that TATA box is a little bit further away from the transcription start site, being found at -25 nucleotides or 25 nucleotides upstream of the transcription start site relative to the 10 nucleotides upstream that the prokaryotic TATA box was found. The TATA box in eukaryotic cells also serves as the site where the double helix will begin to open or create the transcription bubble so that transcription can be initiated, again, because the TATA box, that TA-rich sequence, is easier to separate than a GC sequence would be because of the relative low number of hydrogen bonds holding the double strand together. Also similar to prokaryotic cells, we're going to have a protein that will bind to this TATA box to start the process of initiation. In prokaryotic cells, that protein is the sigma factor. In eukaryotic cells, that protein is termed the TATA binding protein, or TTB, and that TTB is part of a 14-protein complex known as TF2D. Nomenclature: TF stands for transcription factor. The Roman numeral 2 refers to the fact that we're talking about polymerase II, which is responsible for transcribing mRNAs, for example. And D is a letter that simply denotes the order in which this particular transcription factor happened to be identified. TF2D contains the TATA binding protein that will bind to the TATA box of the promoter sequence. This will initiate a cascade of the binding of other general transcription factors, including TF2B, TF2F, TF2E, and TF2H to assemble a multi-protein complex, including RNA polymerase II, that will all gather around that promoter site of the gene that's meant to be transcribed. TF2H is the last general transcription factor to bind to this complex at the promoter site because TF2H is responsible for transferring phosphate groups from ATP to a tail on RNA polymerase II. The transfer of these phosphate groups jumpstarts the process such that polymerase II will leave the promoter site and begin transcribing the mRNA. I put this image here to demonstrate that it's not only the general transcription factors that are required to initiate transcription in a eukaryotic cell. We see in the promoter region, TATA binding protein is part of TF2D. The other general transcription factors about which we spoke, as well as RNA polymerase II. However, there are a large number of other transcription factors that are specific to any given gene. These are known as general transcription factors because they're responsible for transcription of all genes that depend upon RNA polymerase II, whereas these other proteins are responsible for transcription of specific genes. So these transcription factors, called transcriptional activators, are also responsible for associating with the polymerase and initiating transcription in a eukaryotic cell. Again, a much more complex process. The second step of transcription is elongation. This is the same image we saw when discussing prokaryotic elongation, because the process is much the same in eukaryotic cells. A single strand of the DNA is used as a template, as incoming ribonucleotides are paired in a complementary fashion to create the newly synthesized RNA transcript. While the process is much the same, as you might imagine, it's a bit more complex in eukaryotic cells, mostly because of the structure of DNA that the RNA polymerase enzyme must contend with. Core histone proteins: histone protein 2A, histone protein 2B, histone 3, histone 4, and there's also histone 1. The DNA wraps around this core of proteins, all of these histone proteins, to create that euchromatin that must be disrupted if RNA polymerase is going to be able to access the DNA template that it must read in order for transcription to occur. This is an energy-dependent process that requires ATP to move the histone proteins away from the DNA backbone such that the DNA bases can be accessed by RNA polymerase. Just like initiation and elongation are more complex in a eukaryotic cell, termination is also a bit more complex. We'll speak more about this later when we talk about RNA processing. However, there is no official termination signal sequence in the DNA of a eukaryotic gene. Rather, RNA polymerase II simply falls off of the DNA template when the RNA that it is transcribing is cleaved. And indeed, there are cleavage signals encoded by the DNA that we'll speak about next class period. Another complexity in eukaryotic cells relative to prokaryotic cells is the structure that genes themselves have. This is the image that we saw in the last video showing gene A and gene B with their adjacent promoters being on either side of or on either strand of the DNA. Note, however, that there is some DNA that intervenes between gene A and gene B. This is what many people call spacer or junk DNA. This is depicted in a figure here showing you that the amount of junk or spacer DNA in a prokaryotic cell is small relative to that spacer DNA shown in gray that you might find in a human eukaryotic cell. The other thing that you'll note is that while there is spacer or junk DNA between individual genes in a eukaryotic cell, there is also spacer or junk located within the genes of a eukaryotic cell. So there are gray regions here that indicate DNA that, while it will be transcribed, will not code ultimately for a protein. Finally, the process of transcription in a eukaryotic cell is spatially separated from the process of translation. That is because transcription will occur in the nucleus of a eukaryotic cell. An mRNA that is produced will be moved to the cytoplasm where the process of translation will take place. Those two processes, transcription and translation, are not separated in space or time, in fact, in prokaryotic cells, because the DNA itself is located in the cytoplasm. So even while transcription is still occurring, the process of translating an mRNA that isn't completely finished can actually begin. So because a eukaryotic cell spatially and temporally, meaning by time, separates those two processes, things can happen to an RNA prior to translation. Those things are what we call RNA processing. So RNA processing is something we'll speak about next, and it occurs in 3 separate steps in a eukaryotic nucleus prior to an mRNA being transported from the nucleus to the cytoplasm for translation.